So hello everyone. Today we have the pleasure to host Claire Sergent. Claire Sergent is a professor of cognitive neuroscience at the University of Paris Descartes. She's the co-director of the CogMaster program, which is this excellent master of cognitive science in Paris. She's been trained as a biologist at the École Normale Supérieure de Paris. In 2005, she completed a PhD under the supervision of Stanislas Dehaene. And then she did a series of postdoc with Garen Fries, Catherine Talon-Baudry, Lionel Nakache, and she started her own team in 2015. The constant goal behind Claire Sergent's work is to unravel the neural basis of consciousness. Her initial work aimed at characterizing the neural dynamics that accompany conscious access, and more recently she's turned to explore how conscious processes integrate with other processes like perception and decision making, and how they are all interacting but nevertheless separable. So I think this is what she's going to present today. So thank you very much, Claire, for being there today, and the floor is yours.
Okay. Well, thank you very much for inviting me. So indeed, I'm going to give you an overview of what we're doing now with my team, which is trying to distinguish the neural mechanisms of conscious access from sensory processing and decision-making. So first, I want to put conscious access in the general context of the endeavor to understand consciousness. So consciousness is first a term of human language that we use to try to make other people understand what's going on in ourselves. It's naive psychology, and it's very polysemous. So it refers to self-consciousness, reflexive consciousness, phenomenal consciousness, all different themes that can be tackled scientifically. But here my goal is to try and understand this little process here is how we become conscious of a specific content, sensation, thought, etc. at any one time during wakefulness. And the idea is that understanding this little mechanism here will help us first try to understand the difference between different conscious states, but also might reveal how these different aspects of the richness of our conscious life can arise. So to give you a more direct idea of what I mean about conscious access, I like to give this little demo. Maybe you've seen. Right. So if you look at you will see that there will be a little green dot flashing here in the center. I'll ask you to fixate this dot and just look at what happens. So while you're fixating this little dot here, at some point you might experience that one or several of the other dots are going to disappear. And what's interesting is that they don't disappear from the screen or from your retina, they disappear from your conscious access. So this is one of several examples that you can actually have a stimulus that enters the brain, but that can be either processed consciously or processed without this impression that you've had access to this information. So this makes the question quite simple. What's the difference in your brain when the same information is processed consciously or non-consciously? If we understand that, we might have a key to reading out what's going on. Sorry. I was supposed to have a photograph of a patient, but it doesn't matter. So understanding that in healthy subjects might also give us a key to be able to diagnose conscious access in patients that do not communicate. Try to read out from their brain activity if they have these mechanisms of conscious access. Right. So what do we know now about the mechanisms of conscious access? Well, we have like several decades of research that have contrasted brain activity when you become conscious of a stimulus versus you don't for exactly the same external information. So if we try to make a synthesis of what has been found, what are the stages of information processing that correlate with conscious access? We can think of sensory processing, we can think of stages of top-down reamplification, of sustained activity within the global network of areas. What stages correlate with conscious access? Well, if we look at the literature, actually all these stages correlate with conscious processing in one experiment or another, including pre-stimulus activity. So we're now at a turning point where we need to go from what correlates with conscious access to what are the signatures of conscious access, really the mechanisms that correspond to conscious access per se. And for that, we need to dissociate conscious access from upstream events. So for example, fluctuations in pre-stimulus activity that might favor conscious access, future conscious access, even before the stimulus, and also the strength of sensory processing that might influence whether you become conscious of a stimulus or not. That's the first thing. And the other aspect, on the other hand, is we need to dissociate conscious access mechanisms from downstream events. So when you become conscious of a stimulus, on top of that, you become able to make more activities on this stimulus, perform a task, for example. Can we dissociate conscious access from these downstream events? So that's really my current research program, and I'm going to give you examples of what we're doing in these two, on these two topics. So first, conscious access and sensory processing. So if we want to detail what are the different options that that are now on the market about the relationship between conscious access and sensory processing, we have first an early and local view that suggests that we become conscious of a stimulus during the buildup of sensory information within sensory areas, and more specifically, as soon as we have recurrent activity going on between sensory areas. Another view is what we might want to call the latent global view, states that we can only become conscious of a stimulus once sensory representations are shared within a broader network of areas, critically including supramodal areas in the frontal and parietal lobes that can maintain and flexibly process this information. And that's and you will have recognized here the global workspace proposition, and there are other higher order propositions of consciousness that that also would adhere to this latent global view. So how can we disentangle these two propositions? Well, we've seen that if we only make a contrast between conscious and non-conscious processing of the same stimulus, we find evidence for both views. So we thought, how can we decide between these two options? Maybe by considering that in the early and local view, conscious access needs to be time-locked to sensory input. It's really sensory processing that makes you conscious of the stimulus. In this view, it's different. We might consider that conscious access is rather time-locked to this second event here, top-down reamplification, the event that allows the general sharing. So we thought maybe we can manipulate this second phase here. Maybe we can manage to flexibly trigger this second phase at an arbitrary time after the initial phase. And so our prediction was that we could trigger retrospective conscious perception, so that even in situations where the initial sensory processing is not strong enough for the subject to become conscious of the stimulus, if we trigger this second phase and an arbitrary time after the presentation of the stimulus, we can make people conscious of this past stimulus. So we first tried this in behavior, and we implemented a very classical Posner cueing task where people are fixating here, and we present a Gabor patch on the left or on the right at threshold. And instead of just having cues that occur before the target, we also have cues that can occur after the target. And they attract the subject's attention either to the location of the target or to the other location. So if we look at the results, here is time, and the gray area is the moment where the target was on screen. If you cue attention before the target, as here, we recover the classical Posner cueing effect. That is, if you attract attention to the future location of the target just before the target appears, you have a massive boost in the perception of the target. So what happens if you do that after the target has disappeared? Well, if you look at the literature, the assumption was that after the target has disappeared, you cannot change its perception anymore. But when we tested it, we discovered that, yes, you could still change its perception. And even 400 milliseconds after the target has disappeared, if you attract the attention of the participants to the past location of the target, you still improve their ability to report the target, its orientation, whether it was presented or not, etc. So since then, we played around a lot with this protocol. We've shown that you can improve orientation discrimination, detection D prime, subjective visibility, and also more recently extended this phenomenon to audition. So now what happens at the neural level? When we perform these retro cueing protocol. So we performed an MEG experiment using this time auditory cue to orient voluntary attention towards the past location of the target or the other location. In this same protocol. The reason why we chose auditory cues when we moved to MEG experiment is that we wanted to avoid retroperception to tackle the same sensory channels as perception itself, so that when we see the influence of the cue, we know that it's really a top-down influence. But otherwise, the protocol is the same. And I'm going to show you here the results we obtained in looking at the activity in the primary visual cortex. We wanted to see how these cues influence the activity in the visual cortex and how this interacts with becoming conscious of the stimulus or not. So first, let's look at the control condition where the cue comes before the target. So when the auditory cue comes before the visual target, when we look at activity in the visual cortex, we see that the auditory cue nevertheless boosts visual activity even before the target. So that's a nice effect of attention, since here the auditory cue has a visual meaning, it still increases the activity in the visual cortex even before anything visual happened on the screen. Okay, and now after the target, we can look at what activity in the visual cortex correlates with seeing the stimulus in red versus not seeing the stimulus in blue. So in both cases, the stimulus is the same, but at the end of the trial, either people told us that they've seen the stimulus or not. And you see that it makes a big difference. When people see the stimulus, we see these two bumps of activity in the visual cortex, and one around 200 milliseconds and one around 500 milliseconds. And we assume that this first bump here might correspond to rather local activity, recurrent activity in the visual cortex, while the second bump might correspond to a more general communication between the primary visual cortex and higher level areas. So these reamplification that we were talking about. So now what happens if the cue occurs after the target? Do we manage to reactivate V1 as we assume we do? Well, it seems that yes, if we look at activity when the cue here comes after the target, the auditory cue, we see now 3 bumps. The first bump 1 is still there, and we see 2A and 2B. 2A corresponding to the having the same latency as this one, so it's the target triggered reactivation of the visual cortex, and 2B might be the cue triggered reactivation of the visual cortex. So now we seem to validate the idea that indeed, even when the cue comes after the target, it reactivates the visual cortex, not particularly surprising, but now we want to see how it interacts with conscious access. And here we have to consider that probably these curves here are a mixture of trials. Trials where the target would have been seen anyway, because we're at threshold. On some trials, people would have seen the stimulus anyway, and trials of genuine retroperception, trials where initial activity was not strong enough for the subject to become conscious of the stimulus, but the cue rescued the activity. This reactivation rescued the activity, and you became conscious of the stimulus because of the cue. So can we disentangle these two types of trials? Well, we reasoned maybe we might just want to sort the trials according to whether this target triggered peak is higher than the cue triggered peak versus the other. And the difference could be as small as we want. We just wanted to see whether this would allow us to sort out trials where it's the target that triggered conscious access versus it's the cue. So if we look at trials where 2A is bigger than 2B, not very surprisingly, we see that indeed we managed to have trials where 2A is bigger than 2B, but more interestingly, we see that these are trials where the initial activity was really strong. And note that this period here is independent from our sorting procedure. More interestingly, what happens if we look at trials where 2B, the cue triggered activity is bigger than 2A? Well, we observe that actually 2A is absent, and strikingly, initial activity is not different from the activity we would observe in not seen trials. So this seems to validate the idea that this procedure does trigger on some trial retroperception. So indeed, trials where people should not have seen the stimulus based on initial activity, but we nevertheless manage to make them see the stimulus by triggering reactivation with the cue. So this seems to validate the idea that indeed it's this reactivation of this, it's the triggering of this late activity, this late sharing of sensory information with a broader network that seems to be the trick, to be the mechanisms of conscious access itself, and that sensory activity, initial sensory activity is pre-conscious in a strong sense, that the presence of this sensory activity cannot in itself predict whether you'll become conscious of a stimulus or not. It's really this later activity that seems to correlate with that. So another interesting prediction we can make in this perspective is that if conscious access is not necessarily tied to sensory processing, well, it might also be very flexible in content. Let me explain what I mean by that. So let's imagine now that we have a sensory input that is processed up to this stage, but then we manage to mask the low sensory features of this representation, for example, using masking procedures. If we use retro-cueing, can we still manage to reactivate only the high-level features of the stimulus, since it's the only information that remains in the brain? And in that case, we might become conscious of the stimulus, but only the higher level attributes of the stimulus. So in other terms, can we make people perceive abstract representations of a past stimulus detached from sensory features? So that's what we tried to do with my former PhD student, Daphne Rimsky-Robert. So in this experiment, we mask visual words, and after their presentation, people hear another word that can be congruent with this visual word or incongruent. And what we observe is that indeed congruent auditory words will improve word identification. They will improve detection sensitivity. People are more able to say whether a word has been presented or not when after that we present an audio word that is congruent with this previous word. But strikingly, this retrospective word has no effect on the ability of the participants to discriminate the case in which this visual word was written. And this results in a drop in the correlation between the ability to detect the stimulus and the ability to say whether it was uppercase or lowercase. And likewise, we can show that when people can report the masked word in the congruent trials, they are very bad at saying what the font was, what the case was. And interestingly, we show similar results with spatial position. So the retrospective semantic context here, auditory context here, make people more able to say what was the hidden word, but they are unable to say whether it was presented above or below fixation. Right, so this series of experiments seem to suggest that indeed there's a nice dissociation between sensory processing and conscious access mechanisms, to the point where we can consider that these conscious access mechanisms are a very general purpose mechanisms for conscious access that can work on sensory informations, but likewise can work on more abstract informations. Okay, so now, how much time do I have left, please? Sorry.
20 minutes.
Okay, cool. So now, in the second part, I'd like to talk about the other side of the story. How can we dissociate conscious access from downstream events, for example, task performance? So in this perspective, we considered the idea that there might be specific dynamics associated with conscious access, meaning discontinuous dynamics. So we already had hints that it might be the case back in 2005 in the attentional blink protocol. And this has been confirmed in other protocols, for example, in masking by Antoine Delcu and Stanislas Dehaene, and by Sid Quider as well. So there were indications in the visual domain that becoming conscious of a stimulus might be associated with discontinuous dynamics in brain activity. So we wanted to look into that further by generalizing this observation, looking at whether it was a general signature. And also you will see by formalizing a bit more what we meant by discontinuous dynamic. So first, in what sense do we want to generalize this signature? So as I was saying, we only observed that in visual processing and also in protocols where the stimuli themselves have sharp transients. So in this experiment, we wanted to see whether these discontinuous dynamics could still be observed in a very simple protocol, audition around threshold with no masking, no fast transients. Another way we wanted to extend these results is to see whether we could find these discontinuous dynamics with and without a task, because usually in these experiments, we ask people to report the stimulus, but the correlates that we observe when people report the stimulus might correspond to task-related activity rather than conscious access itself. So here's the protocol that we came up with these two objectives in mind. So people were listening to a continuous threshold equalizing noise like this. And within this noise, we embedded two possible vowels in French, A and E, at different intensity levels relative to the noise. So that's why you see minus 13 dB, etc. So from minus 13 dB to minus 5 dB, with these two values corresponding to the auditory threshold. So this, the level of intensity where people heard the stimulus around half of the time. And there were also trials where no sound was played. And people performed two sessions, counterbalanced between participants, and there were 20 participants, by the way. They performed an active session in which, at the end of each trial, they had to report which vowel had been presented and to report on a scale between 0 and 10 how audible this vowel was. And in a passive session, they were the stimulation was exactly the same, but at the end of each trial, instead of reporting the vowel, they had to respond to a quiz test or to different visual tasks. Okay, so as I was saying, one of our goals was also to try and formalize a little bit more what we meant by discontinuous dynamics. And so these are simulations. We thought what we want to contrast is first unimodal dynamics. So brain processes which increase in activity when stimulation strength increases in a gradual manner, meaning that at each level of sensory input, all the trials organize around a unimodal distribution around just one mean. So that's the classical distribution. And we want to contrast that with bifurcation dynamics, in which when you are, when stimulation is around threshold, you have two types of trials. On some trials, activity remains at baseline, and on some trials, activity reaches a high state, and you have a group of trials around this higher mean here. And if you tilt your head, you will see the distribution of activity at threshold stimulation seems to be bimodal. There are two bumps here. Okay, and so we thought, how can we disentangle these two possibilities, knowing that our idea is that if you have processes that show bifurcation dynamics, they might correspond to non-conscious processing versus conscious processing. Well, if we do the classical analysis and average activity across trials for each stimulation level, well, the response curve is going to be very similar in these two dynamics. What we need to do to capture this distinction is to look at variability of the activity across trials. And indeed, the prediction is very interesting if you look at the profile of variability as a function of stimulation strength, because for unimodal dynamics, you predict monotonous increase in variability with increase in activity and increase in stimulation strength. While for bifurcation dynamics, you predict a very peculiar profile where you have a burst of variability at stimulation threshold, because you have this split between two types of trials. And as you move away from threshold, you either go to all the trials are grouped around high activity, or all trials are grouped around low activity, and in both cases, variability gets down. So can we just simply see whether some stages of processing show these bifurcation dynamics with this signature in the variability? So that's what we looked at first in active listening. So these are the evoked potentials for the strongest sound, where we see the classical auditory N1 and then a nice long P300 in response to the auditory stimulus. If we look at the profile of mean activation around a group of electrodes here that capture both the N1 and the P300, we see that indeed activity increases with stimulation strength. Here it's negative activity, so the intensity of this negative activity increases with stimulation strength. But we've seen that this average cannot tell us much about the specific dynamics behind these activations. So let's look at variability across trials. And during the initial phase, up to 300 milliseconds after the presentation of the stimulus, we see that the profile of variability seems to have a monotonous increase that seems to be like unimodal dynamics. And then later on, we see very clearly this specific pattern that seems to be specific of bifurcation dynamics, where we have a burst of variability across trials around threshold stimulation. So we see the same profile when we use more refined analysis techniques using multivariate pattern analysis. And so now we can look at activity in more details, so trial by trial activity, and look at the predictions we make on the distribution of activity across trials for unimodal model and bifurcation model. So with increasing stimulation strength. So here's the prediction for the unimodal model. So activity increases, the mean activity increases. Maybe there is a widening of the Gaussian distribution. So an increase in variability, but monotonous. In the bifurcation model, we predict that for intermediate stimulation strength, we'll go in, we will have a distribution with 2 bumps, and then again slide into more unimodal distributions. And here is the observed activity for one participant, and we see something similar when we put every participant together. Observed activity at the late processing phase, and we see that indeed there are nice indications that for stimulation around threshold, we don't really have one unimodal distribution. It seems that there are 2 types of trials. So what we did is we fit our 2 possible models, unimodal model and bifurcation model, to our trial by trial activations as recorded in EEG. And here are the results at the group level. So 0 is the moment where the stimulus has been presented. And we see that indeed the unimodal model seems to explain the data better up to 200 milliseconds. And then we switch to bifurcation dynamics. So next, we wanted to see how this fits with participants' report of their own conscious perception of the stimulus, because we're still in the active listening session. And what we did is to use this model to try and predict the response of the participant, namely based on trial by trial activity at different time points. This bifurcation model would give us a bet on whether on this trial activity was in the high state, so probably conscious, or in the low state, so probably unconscious. And indeed, this procedure allowed us to predict accurately the actual response of the subject, whether they had heard the stimulus or not. So the most interesting part is obviously to try and compare these signatures of conscious access in the active session with what happens when people do not make a task on the stimulus. And many authors consider that this P300 activity that here seems to correlate with the conscious access actually is only task related. If it's the case, then it's when people do not have to make any task on the stimulus, this should disappear. Conversely, if this late activity corresponds to conscious access, it should remain in the passive session. So what is the case? Does it remain or does it disappear? Well, neither. Actually, what happens is that during the passive listening, we still have strong activity during the late time window up to even later than 600 milliseconds after the stimulus, but this late activity is not the P300. It's a different topography. So what we reasoned is that probably this late activity here might correspond to spontaneous conscious access. And this spontaneous conscious access might be actually included in the P300. And that's, this interpretation is actually validated by performing a cross-classification analysis, where we see that indeed passive activity here is included in the active P300 that we observe. That might be easier to understand when we look at the source reconstruction of our effects. So in the active session, here is the network of areas that we see activated in this late time window, where we have obviously temporal activations, and these are auditory stimuli, but also parietal and frontal activations. When we look at activity at the same time window, when people are passively listening to the stimulus, we still, it seems, so these are EEG reconstructions, but it seems that we still have a network of areas that is activated, so temporal, frontal, and parietal. And interestingly, the difference between the two is just related to executive area and more precisely motor planning areas. So we still have to confirm this framework using fMRI, but one possibility is that if this network of area here reflects a global workspace that makes a sensory stimulus conscious for a particular task, well, this network might correspond to a global playground. So a network of areas that allows to maintain up to 600 milliseconds and circulate sensory information, but not necessarily for a particular purpose. And the difference between the two might be whether you connect this global playground to executive areas or not. Still under investigation now by performing fMRI and intracranial recordings. So last part is to look at the dynamics of activations in this passive listening condition. And indeed, we observe during this late time window or signature of bifurcation dynamics. And at this stage, we thought, okay, that's cool, we've made it. And then reviewer 2 made an interesting remark. It was annoying initially, but then we thought that it was a genius idea. They said, okay, you're telling me that this late activity here during passive listening corresponds to people becoming spontaneously conscious of the stimulus, but how do I know it's the case? How do I know that they are becoming conscious of the stimulus during these high activity trials? And this seems to be difficult to address since people are supposed to be passive on the stimulus. But then we thought, oh, but we had mind-wandering probes during these passive listening sessions. So it means that it's 10% of the trials. No, I think it was more than that, 20% of the trials where participants, at the end of the trials, we asked participants, what's in your mind now? And we gave 4 options. I'm concentrated on the quiz questions because they were doing a task. It was not on the stimulus, but they were doing a task. Or I'm in my thoughts, or I'm drowsy, I'm sleeping, or I have the sound in my mind. And we thought maybe based on trial by trial activity and or bifurcation model, we might be able to predict these mind-wandering responses. We might be able to predict whether they respond to the mind-wandering probe by saying that they had the sound in their mind. And indeed, that's what we observed. So we were able to predict their responses, whether they had the sound in their mind, using activity, using trial by trial activity before the mind-wandering probe. So to conclude, it seems that these retroperception protocol allows us to disentangle upstream events from conscious access events in a very strong way by desynchronizing what belongs to sensory processing per se and what belongs to conscious access mechanisms. And we have the hope that these bifurcation dynamics that seem to generalize to the auditory domain and to very simple protocol, and importantly, that seem to generalize to the absence of a task, might help us reveal signatures of conscious access that we can read out from brain activity without people being, without people telling us whether they are conscious of the stimulus or not. And one obvious application that we're trying to investigate now is to use this kind of protocol to try and probe conscious access in non-communicating patients. So to conclude, I'd like to thank all my collaborators on these studies and present you my current team, and I'm happy to take questions. Thank you.